going to look at different types of vaccines. So based on the method using which vaccines are prepared, there are different types. So let's look at how vaccines are made. So vaccines are made either by weakening the organism. So in this context, we need to remember the word attenuation, whereby the organism loses its pathogenicity, however, does retain its antigenicity. Second thing is by inactivating the organism. In other words, by killing the organism. So there is no question of pathogenicity here. However, antigenic properties will still be retained. The third way by which vaccines can be prepared is by inactivating a toxin. So toxin is nothing but a poison which is prepared from the bacteria. So by inactivating this toxin, we can produce what we call as a toxoid, and this toxoid can provide protection against this toxin if the person is exposed to the toxin in future. And lastly, which is very commonly used these days, is using a part of the virus or the bacterium. So using a part of the cell wall or the capsule of the microorganism to produce a vaccine. These are the techniques by which vaccines can be prepared. On the basis of these methods of preparation of vaccines, we have different types. So the types here are live attenuated vaccines, whereby we weaken the organism; inactivated or killed vaccines, wherein the organism is killed. Please remember, both in the live attenuated and the inactivated variety, the antigenic properties are still retained. Third variety is toxoids, when we inactivate the toxin produced by the bacterium or virus. Point to remember here is toxoids are effective only against toxin produced by the bacterium or the virus and not against the organism itself. The third variety is polysaccharide and polypeptide or cellular fraction vaccines, whereby I mentioned that use of part of the organism. So either the capsule or the cell wall can result in different types of vaccines like this. And the last variety is surface antigen or recombinant vaccines, wherein we just take the antigen from the organism, not the whole organism or not a part of the organism, just the antigen, and then we make it into a recombinant variety in order to produce a vaccine. So we are going to look at different types of vaccines and examples of each of them in the next slide. So let's move on. So live attenuated or avirulent vaccines, as they are known as, so we need to understand that virulent pathogenic organisms or organisms having the capacity to infect and cause infection and disease, they are treated. In other words, they are subjected to chemical form of treatment or biological form of treatment so that they become attenuated and avirulent, whereby they lose their pathogenicity but they still remain antigenic. So what happens basically is these organisms have lost their capacity to induce full-blown disease. So actually we are not injecting anything other than the organism. We are injecting the organism. However, this organism does not have the capacity to induce full-blown disease in the individual. However, once it enters into the body, it will promote the immune system to cause the immune reactions to form antibodies, because there is antigenicity and immunogenicity which is retained in these vaccines. The next variety is inactivated or killed vaccines. So by heat treatment or chemical treatment, organisms are killed or inactivated. So there is no question of pathogenicity here because the organism has already died. However, this organism still remains antigenic. What is important to remember about inactivated vaccines are they are usually safe. However, they are less effective than the live attenuated vaccines. Reasons I think are quite logical to understand. When we introduce a live attenuated vaccine in a human body, the organism is going to multiply, whereby immunologic memory is built up. Whereas when we inject or introduce a killed vaccine into the human body, what will happen is the immunogenicity or the antibody formation is proportional to the amount of dosage of the vaccine we give. Because once the vaccine is given or once the organism is introduced, it's not going to multiply within the body. The number remains the same. That's why if we come to maintaining the immunological memory or talking about maintenance of the immunity over long periods of time, live attenuated vaccines are much better than the inactivated ones. The next variety is the toxoids, whereas I said earlier, what is done is the exotoxins of some bacteria are detoxified, whereby again the principle remains the same. The toxin will lose its toxic properties, however, it will retain the antigenic properties. So what happens now is the antibodies which are produced in the body as a consequence of this toxoid administration will neutralize the toxic molecule or moiety produced during the infection, and it is not going to act on the organism, the bacterium or the virus itself. So that's very important to remember. Toxoids are effective only against the toxin produced by the specific organism. Right. The next one is polysaccharide and polypeptide or cellular fraction vaccines. These are the vaccines which are prepared from extracted cellular fractions. So either it would be the cell wall or the capsule of the organism from which the antigen is derived and then it is introduced into the human body. The last variety which we are going to speak about today is surface antigen or recombinant vaccines. Now these vaccines are prepared by the process of cloning. So what happens is the common thing which is used is for hepatitis B surface antigen. So what is done is the gene of this hepatitis B surface antigen is cloned in the yeast cells and it is expressed in these yeast cells. The new variety of hepatitis B surface antigen which is produced in the yeast cells now is then used for preparing the vaccine against hepatitis B. So this is an example of surface antigen or what we call as recombinant, because this gene of the antigen is modified to produce another antigen which has got the property to induce antibody production. So in a nutshell, now what we are going to look at is a table which shows us examples of the different types of vaccine. So we have live attenuated, killed or inactivated vaccines, toxoids, cellular fraction vaccines and recombinant vaccines. So here you can see down the examples of each of them. So the vaccine against tuberculosis, BCG, is a live attenuated vaccine. The other examples are oral typhoid, oral polio. Why the word oral here is important is based on the different routes of administration, the types of vaccines are different. So you can find oral typhoid as live attenuated, whereas typhoid vaccine as a killed vaccine. So there are certain vaccines which may be available as live attenuated or killed vaccines depending on the way in which they are prepared. Then you have live attenuated vaccines for plague as well as killed vaccines for plague, and you can see other examples like measles, mumps, rubella. For polio, you have oral polio as live attenuated, whereas Salk polio or injectable polio as killed vaccine. So these are the examples of live and killed. There are two toxoids as of now available. One is diphtheria and the other is tetanus. The examples of cellular fraction vaccines are meningococcal polysaccharide vaccine and pneumococcal polysaccharide vaccines. So we have vaccines against meningococcal disease and pneumococcus, the organism, which are cellular fraction vaccines which are made from the cell wall and the capsules of the organism. Another example of cellular fraction is hepatitis B polypeptide vaccine. So for introduction against hepatitis B, we have the polypeptide variety of the vaccine. The last type of vaccine which we have looked at today is the recombinant vaccine, and the best example of which is hepatitis B vaccine. So the surface antigen of hepatitis B, which is the gene of which is cloned in the yeast cells, is then ready for administration in humans. So that's all in a nutshell about the different types of vaccines. In the next session, we will be looking at what are the different routes of administration of vaccines. Thank you very much for watching. Do subscribe. Have a great day. Bye.